board or whatever, the sign that was like, find a tick, send it here. And in the early years, they even used to get the pathogen identifications and just tell people like, oh yeah, that tick was infected with Lyme disease, that tick wasn't. Definitely that you can't really do that sort of thing anymore. There's too many liability risks for non-doctors to be telling people that sort of thing. Yeah, and then we had a real active internet presence, and while I was running that program, I would post the monthly tick data for across the state, and kept it all up to date, month to month, up to date, and we had a website and stuff, and so we ended up having lots of people sending in ticks from out of state because their own states didn't support any kind of free identification program. So, but yeah, outreach. All right, so yeah, I'm going to talk really fast about tick-borne diseases. That delay at the beginning kind of caused some problems, but yeah. So here's the topics. I'm going to mostly talk about Lyme disease and human granulocytic anaplasmosis, just because those are Lyme disease is 99% of tick-borne disease or vector-borne disease at all in the U.S. And then human granulocytic anaplasmosis is probably number 2. So yeah, anyhow, tick-borne diseases are seriously underreported. Everyone knows this. Even the CDC knows this and says, yeah, there's at least 300,000 cases of Lyme disease a year. But official reports are typically 30 to 35,000 cases per year nationally. So there's at least 10 times more cases of Lyme disease each year than what are actually getting captured by the surveillance system set up by the CDC. And this most recent study where they looked into insurance records and doctors' diagnoses and that sort of thing, found closer to 500,000 cases per year in more recent years. So it really is a lot of cases that go underreported or completely unreported. Now, most of these people are probably getting casually diagnosed by their doctor. They're getting their antibiotics or whatever, but they don't meet the strict CDC case definition. So, if we consider this, this is a little old, it's 2011 data. But, in 2011, there were 33,000 official cases of Lyme disease, when really there should have been, probably more like 480,000, 475,000 official cases of Lyme disease, which, at least bumps, certainly salmonella is super underreported too, right? But, definitely Lyme disease is a more common issue than HIV, new HIV cases or new syphilis cases. So yeah, it's people are finally coming to terms with the idea that this disease is very common. Similarly, we really know very little about human granulocytic anaplasmosis, HGA, but those few studies that have looked at it in sort of general populations have also found high levels of exposure. Now, just because someone is exposed doesn't mean they're going to get sick. And then there's also non-human infectious versions of the bacteria that cause HGA. There's humans can get the one that's carried by mice, but the one that's carried by deer, and transmission between deer and ticks, that one does not affect humans, and so we won't get sick. But you would still develop an immune response against it. If you're bitten by a tick that injected into your body, you don't get sick, but you still have the it. So anyway, there's likely a lot more anaplasmosis out there than people are recognizing too. They found that 11, it's a tiny N, right? 18 out of 159 people had antibodies against anaplasma, but still, that's 11% of people. And this is in Westchester County, New York, which is a county of like 1 million people, right? And so, you know, I don't think you could say, well, let's extrapolate that, you know, 100,000 of them had anaplasmosis. You can't really say that, but still, this is clearly a highly underreported disease, I think, based on this data. So the reason the CDC case test designation is so strict is that they require, in order for something to be officially registered as a case for the CDC, you need an EM rash, you need either a positive ELISA or a positive immunofluorescence assay, and you need 5 positive bands on a Western blot test. And so, you know, people develop the EM rash at like 3 to 30 days post-bite, right? And so that's fairly early. That's when, you know, symptoms are coming on too, usually. But they're never going to get 5 positive bands on a Western blot in like 3 days. You know, it's going to take, you know, 4 to 6 weeks probably for them to develop a full-on immune response. And so you get this sort of complication where like most patients aren't ever going to fall into this bucket of having both the EM rash at the same time as having the Western blot, right? So that goes a long way towards explaining the problem with the CDC numbers. All right, so yeah, deer ticks have a bunch of, are associated with a bunch of emerging diseases. We find new tick-borne diseases all the time. Maybe not all the time, but, you know, fairly often, a couple times a decade. There are new ones that are coming out, you know. Let's see. Human granulocytic anaplasmosis, first discovered as a human infectious agent in Minnesota. Ehrlichia like agent, first discovered as a pathogen in Minnesota. Lyme disease caused by Borrelia mayonnae, first discovered in patients in Minnesota. So there's certainly plenty of overlap with Wisconsin too. I mean, it's all the same ticks, but all of those diseases are originating in this same species of tick, right? So there's something going on with this tick, probably has to do with its density on the landscape, and plus it's like multi-host, you know, it's variable host preference and all that sort of thing. I don't think anyone really knows, honestly. But they can carry a lot of different diseases, and we continue to discover new ones. So which one's the deer tick? Yeah, it's this one here in the upper left. Though this one here is also, you know, gonna look identical, basically the Western black-legged tick. So this is missing the new one, the Asian longhorn tick. I haven't put that on this slide yet.
In your folders, I hand out about Asian longhorn.
I've got a little bit at the end of the talk about them, but it's just not on here. They're not really that well. You know, they transmit a bunch of diseases in Asia, but we still don't really know what they're going to be transmitting of our native diseases here in the US. It seems like they're probably not going to be transmitting Borrelia, though, Lyme disease, so that's good. Yeah, so, right, Ixodes scapularis can transmit a bunch of different diseases. Here's a sort of simplified list. They can also transmit like dog heartworm, deer tick virus, which is basically the same as virus. These are the major ones. So I'm going to talk about these two diseases really quick, just so everyone's familiar. Also, as both Scott and I have mentioned, these ticks have been like massively expanding their range, right? So in the last 25 years, I think there's even a newer version of this map out that, at least in Minnesota, shows all of the northwest portion of the state would also be red. But yeah, so these ticks have been greatly expanding probably everywhere where anyone here lives and works. So in that passive surveillance program in Iowa, we were able to show that these ticks have also majorly expanded across the state. Here's a comparison of what they had in that 2015 map. It's not really up to date compared to this one, but yeah. So if any of you guys have been to Iowa, if you've ever been lucky enough to have been to Iowa, it's like 98% corn and soybean fields, right? It's not what you go to and say, oh, this is tick habitats. But clearly these ticks are able to survive and thrive and spread across the landscape. And so there's no one really understands the mechanics of that. But it probably just boils down to the ticks get on deer. The deer moved, between small patches of like abandoned homesteads or something that have turned into these, hedges. And then the ticks fall off the deer and they live in those patches of woods out, in the middle of a cornfield. And so they've been able to really colonize like much of the state at this point. Probably will colonize the whole state at some point. But yeah, so this is all based on that passive surveillance data, right? Here's another map generated from that passive surveillance program data. This shows submissions by town. We only had like people's names, addresses consistently. Sometimes people would say like, oh, I got it at this park, I think. But we mostly just mapped it based on whatever their hometown was. And so you can get quite a bit out of this map in terms of like, where is Lyme disease risk? Well, clearly there's a lot of risk up there in the northeastern part of the state. We've got higher levels of Borrelia infection in the ticks, and then the size of those circles, indicates sort of how many ticks were submitted. Clearly, there's just a ton of them coming out of northeast Iowa and higher rates of infection, as opposed to down here in Des Moines, where we get tons and tons of ticks submitted from there, but not that many of them were deer ticks. So yeah, this is the sorts of data that you can get easily out of a passive surveillance program. You can also get data like this showing trends over time. So this shows the proportion of infected adults and nymphs over time. So the adults is let me see, it's a little confusing graph. Obviously I made this, when I was a postdoc, but yeah, so you get these confusing lines. But anyway, you can sort of just look at the sort of general trend, and clearly like there's been major increases over time in the prevalence of Lyme disease in ticks in Iowa. And we see these same trends everywhere in the country. No one's really sure why that's going on either. But I think it has to do with, density of ticks on the landscape. I think that pushes these numbers up because of like co-feeding ticks. You get one infected tick, one uninfected tick. They're feeding on, say, an uninfected mouse, but that uninfected tick can still get infected by being near that infected tick. So if you have more ticks around, there's going to be more horizontal transmission from tick to tick. That's sort of at least one potential explanation. But so in Iowa, through this passive surveillance program, we saw this trend of increasing, and then let's just end the program there once we know that 30% of the adults are infected. In Minnesota, we have even higher rates of adult and nymphal infection. All right, so anyway, Borrelia burgdorferi.
10 minutes.
Okay, cool. So I'm having a hard time reading it. Okay, Borrelia burgdorferi is a spirochete. It's got this really unique morphology. Early symptoms before 30 days are the rash, and then you can also get flu-like symptoms, but those are extremely vague. That can just be like feeling tired, in some people, or it can be flu-like. It can potentially move on to much more severe symptoms where you get involvement in the nervous system, involvement in the heart, or joints are sort of the classic. So actually different strains of Borrelia burgdorferi, like if you're talking about the European ones versus the US ones, they seem to have different sort of propensities to cause different sorts of symptomology. Like thankfully the American one very rarely causes like direct brain problems, whereas the European one more often causes brain problems. The American one tends to cause more joint issues, which, you know, I would take joint issues over brain damage. But you know, another major concern though is like heart involvement, and there have not been any good studies about, you know, we know that there is cardiac borreliosis and that sort of thing, but no one really knows like how widespread it is, or what kind of like how many years are being lost off of people's lives because of it. But of course, Lyme disease is not considered a fatal disease. Very, very rarely has that kind of, very, very rarely, if ever, causes fatality. So when a tick ingests Lyme bacteria, Borrelia, it sort of lives in the gut of the tick, and then, you know, during the molting process, and then so it's in the tick's gut when the tick is molted to a nymph. When that nymph bites onto an animal, the bacteria actually migrate over to the salivary glands. They actually have to cross the open circulatory system of the tick and get into the salivary glands and then be injected into the host. And so that process of migration over to the salivary glands and injection takes pretty much at least 24 hours. So this is where that, you know, daily tick checks recommendation comes from, you know, is because you can have an attached tick for up to 24 hours and your risks are still very low for transmission. This has been pretty well studied in Borrelia, you know, based on 4 whole studies, which is actually a lot for any kind of tick research. Yeah, by 24 hours, if you only have 1 infected tick on you, you basically have a 0% chance-ish of getting Lyme. So this was all, this and the next slide are reviewed in this paper by Eisen, which is a really cool paper by Lars Eisen, where they review all these studies. And so this is sort of the synthesis of these different studies. Of course, if you have a bunch of infected ticks on you all at once, all bets are off, you know. But you're really, yeah, at 24 hours with 1 tick, you're still pretty safe as long as you properly remove that tick. Now, if we're talking about other diseases, really nobody knows very well what's going on. But with these other bacterial and eukaryotic diseases, it does seem like you have kind of an early window where you're much less likely to get the disease than if you allow the tick to completely engorge on you. And so that 24-hour time point is still pretty good, probably. I mean, it's certainly better than nothing. But you know, yeah, well, yeah. Anyway, if the tick is infected with Powassan virus, it's systemically infected, its salivary glands are already completely chock full, you're going to get that like right away. So there's sort of nothing you can do. If you get bitten by a tick with Powassan virus, you're probably getting exposed to it. So yeah, here's a slide I show in just about every single talk I give. You know, wear your DEET and, you know, check yourself for ticks at least once a day. Doesn't actually have to be DEET. It just has to be an EPA approved repellent, and there are really good DEET alternatives on the market right now, like oil of lemon eucalyptus based repellents are really good, and they last almost as long as DEET. They're as effective and last almost as long, and they smell better. Yeah, so a little bit on Anaplasma phagocytophilum. So the same passive surveillance program, we found this huge number of ticks that were Anaplasma infected when we looked at them. Out of 156, like almost 30% of them had Anaplasma phagocytophilum. But in Iowa, there's not considered to be any, I wouldn't say any, but you know, the health department only gets reports of like 5 or 6 cases of anaplasmosis per year. It's not a reportable disease in Iowa, so we're not really sure. But most likely these again represent this non-human infectious type of Anaplasma phagocytophilum. rather than the rodent transmitted one. It's the deer/tick cycled anaplasma that's not really getting people sick. But, you know, the ticks still have it, and it's still the same thing. Just thankfully, it doesn't affect us. So Anaplasma phagocytophilum is a really cool bacterium, one of my favorites. It replicates inside of cells, so it's an intracellular bacterium. And one of the really neat things about it is that it can actually it'll actually replicate inside neutrophils. And so it's essentially like taking your immune cells, you know, your white blood cells, and just like moving in and, you know, turning them into a little nutrition transportation factory for itself. So unlike Borrelia, Anaplasma phagocytophilum can cause death, but it's very rare that people die of it. In almost all cases, even if this wasn't treated with antibiotics, it would resolve on its own without any lasting consequences. But again, symptoms are super generic: malaise, chills. There's nothing really diagnostic about human granulocytic anaplasmosis. So we get a third to a half of cases are in Minnesota and Wisconsin. So we get a ton of them up in this part of the country, definitely the biggest concentration. And it seems like there's this big northern shift here. People hypothesize that this has to do with like when the larvae and the nymphs are feeding, like their phenology, their timing of feeding throughout the year, because mice don't stay infected with this for more than a couple weeks. Varies with different strains, but they typically are only infectious for a couple weeks. And so you need to have basically this particular window where the nymphs can feed and infect the mice, and then the larvae can feed and pick up the next generation of infection with this bacterium. And so you need to have basically this perfect sort of timing. And so this kind of distribution, if this is in fact the case, you need this like particular phenology for anaplasma to be a problem. This is the kind of thing that actually could be really disrupted by climate change. You know, we might see cases of anaplasma in Minnesota and Wisconsin declining through time, or moving north in the state or something like that. But you do see it quite a bit also in northeastern US. Yeah. So like I said, the bacteria sort of grow inside your immune response cells. Yeah, all right. And just really quick, here's a picture of the Asian longhorn tick, Hemophysalis longicornis. It's a super cool tick because, well, for a number of reasons, but the most interesting thing is that in North America, our ticks are all parthenogenic or parthenogenetic, however you say that. And so that means that they're all females, right? And they don't need no man. And so these also have a super broad host range, like, you know, farm animals and stuff, but they're even on birds and stuff. And so you can imagine, like, if a female feeds on, if an adult tick feeds on a wild goose or something, that goose flies 500 miles, the female gorgeous with blood drops off. It doesn't need to even mate. It doesn't even need another tick there in order to establish a whole population of ticks in this new location. And so, you know, we fully expect these things to spread to their maximum extent that the habitat will allow them to at some point. And so, of course, their habitat also includes Siberia. The ones that we have here, these parthenogenetic ones, they seem to be a little bit more warm weather adapted than the populations where there's both males and females. And so hopefully they're not going to be super cold tolerant like the ones that we see in Siberia that are not parthenogenetic. So hopefully the ones that we see, so hopefully these ones won't make it to Minnesota is what I'm saying. But if you're in New Jersey or Virginia, they're definitely probably already all over the place. And you can get just insane, disgusting quantities of these. I heard of some guys who were like working in upstate New York, like putting in power lines in the forest, and they left their truck running. And so, you know, was generating CO2. And like the whole like bed of the truck ended up like full of these ticks. Not like drifts of them, but you know, they're like all over the bed of the truck. You know, they're like ticks all crawling all over the place because they've been attracted. Because you know they can reach incredibly high densities because they don't need any kind of mating or anything. Yeah. So anyway, this is the most recent map that I have. There's probably a more recent. Oh yeah, this is from a year ago. I'm sure there's more recent maps, but this is a relatively recent map. And this site over here in Arkansas has had these ticks there for a long time, probably from something like a bird flight or something. But yeah, they haven't really spread out of there yet. But most likely it's going to be a broad swath. We don't really know what these ticks can transmit in the U.S., as I already mentioned. In China, though, they've got this disease called severe fever with thrombocytopenia syndrome virus. It's got a new name to it. This one is super duper concerning. Some of the early outbreaks had greater than 50% mortality. And so, you know, we're talking about like on par with like Crimean Congo hemorrhagic fever. Once treatment was a little bit better understood, mortality rate overall is down to like 5 to 10%. But basically, so as of November 2017, there were like about 7,000 cases. And then since then, the government has just stopped reporting this, at least in English. And so there's not really any new available data as far as I can tell on it. But, you know, probably they've got it under control. But it's super unlikely that this would ever get imported to the US. You know, we'd have to have like a new importation of their ticks, but we know that this thing can transmit a bunch of different things in Asia. And so it's going to be really interesting to see, you know, what it's going to transmit here. All right, cool. So I think that's it. Yeah, done. Any questions? I just have to go to the bathroom. Sorry.
Okay. And then we're doing lab separately, right?
Yeah. Oh, okay. I'll go open up.
Okay.